Good evening, and thank you for tuning in to our regularly scheduled program, The Complete History of Science. The year is 1946, and this week we'll be delving into bacteria. So please enjoy this brief documentary about our current understanding of this mysterious organism. Our story begins in 1876, when Robert Koch exposed the anthrax bacteria as an agent of infection, carrying illnesses from one infected person to another, and for a few decades, bacteria remained much of a mystery. It was commonly thought that bacteria was distinct from the higher organisms like us animals, and had a mystical life-giving protoplasm that allowed it to reproduce by dividing. Flash forward to 1928, when Fred Griffith observed the apparent transmission of genotypic changes, converting the bacteria Streptococcus pneumoniae from non-virulent to virulent strains. This transforming factor wasn't discovered until 1944, when the trio Avery, MacLeod, and McCarty identified the pneumococcal transforming agent as DNA. Suddenly, the bacteria's entire internal system was called into question. Did this DNA strand carry genes? Do bacteria have genes? Were they able to reproduce? It still remains a mystery that... Sorry for the interruption. For those of you just tuning in, we've recently received word of disturbing sexual scandal regarding bacteria. We are joined today by the accuser, Jay Lederberg, and the defendant, Escherichia coli, and call me K12. So my understanding here is that Lederberg, you are accusing K12 of engaging in sexual relations? That is correct. And completely inaccurate. I deny the allegations. Well, how did you stumble upon this discovery? It's a pretty bold claim. Well, it wasn't that bold. Dubrose's monograph, The Bacterial Cell, really revealed the flimsy evidence supporting bacteria's supposed lack of sexuality. Salmonella had somatic and flagellar antigens in different combinations, implying the existence of random or natural recombination. I met my mentor Francis Ryan in 1942 and tried to replicate DNA transformation in Neurospora. When that didn't work, I thought, why not just test my hypothesis? And so I tried to model recombination in E. coli. So what was the experiment? I'm glad you asked. My partner Tatum couldn't be with us today, but I actually recorded the experiment, so I'll show that to you now. Using radiation, Tatum mutated our E. coli strain K12 here into the two auxotrophic strains, Y10 and Y24, both with different nutritional deficiencies. Y10 needed to be supplemented with threonine, leucine, and thiamine to grow, while Y24 required biotin, phenylalanine, and cysteine. When we combined these strains in mixed cultures of about 1 billion cells, we observed the growth of 100 colonies, and in those colonies was the original K12 strain. After eliminating transformation through controlled experiments with sterile filtrates, we concluded that this strain arose from a sexual process between Y10 and Y24. It's ridiculous. How did you know I hadn't spontaneously reverted back to my original parental genotype? It's interesting because I actually published a paper on this phenomenon in Neurospora with Ryan. So spontaneous reversion to auxotrophy followed my attempts to model transformation in Neurospora before this. But you'll notice here that I use triple mutants, and the chance of spontaneous reversion for all 3 mutations was vanishingly small. Now that is interesting. K12, how do you respond? Well, I actually brought a few of my lawyers. For instance, I have Mr. Cyril Hinshaw, the famous physical chemist and Nobel Prize laureate. Thank you. My client here has been falsely accused. Bacteria are simply reaction systems. They only adapt in response to their environment to spontaneous biochemical reactions. How else are you supposed to explain the rapid response to nutrient sources? Yes, Lamarckism. Well, it's an interesting theory, this idea of holistic adaptation. But only 3 years ago, Luria and Delbruck had proven that bacteria mutate not in response to its environment, but randomly, disproving this idea of acquired characteristics. And now my study on genetic exchange explains the rapid spread of a favorable trait. Well, that's okay, because I actually brought another lawyer, the microbiologist and Nobel Prize laureate, Jan Zabel. Mr. Lederberg, how can we be sure that these results did not arise from, say, the crossing of nutrients between the mutant strains? Well, I did take careful pains in the experiment to avoid cross-contamination. But to be sure, Dr. Maxwell did help me isolate recombinant strains under the microscope, confirming single cells with parental characteristics. Our paper should be out soon, maybe 1950. Do you know what the implications could be if the scandal was released? You've revolutionized our understanding of bacteria. Now that I have a genetic system, I'd just be used for further experimental analysis. I probably help develop your understanding of virulence and the transmission of antibody resistance, eventually improving vaccines. I know, I should win a Nobel Prize for this. Well, that about wraps up tonight. Thank you for joining us here at the Complete History of Science for a historic moment, and good night.